some slight differences in the plasma membrane of the archaea. We'll talk about those more in chapter 4. But the basic thing to remember is that the archaea are in much more extreme conditions. If you look at the figure in your textbook that corresponds with the figure I'm showing you now in the slide, it's 2.1, some of the environmental adversities that are out there. Look at where the archaea are surviving. You've got your hyperthermophiles. Those are the things that survive at 106 degrees Celsius, and they thrive at that temperature. You also have things that can survive at very high alkaline conditions or very low acid conditions, acidophiles and alkaphiles. Your archaea like really salty conditions, the halophiles. These things that live at these very extreme conditions need to have a more robust membrane. It's still a fatty fluid membrane. It still has a phosphate group that is attracted to the water. However, our central fats, our lipid tails, actually connect to each other, and you get a monolayer in the archaea. It's very interesting. It is one of the reasons when we look at the phylogeny of life here that we see the archaea in their own domain. It's because of that very distinct plasma membrane. But the chemistry of it's still the same. However, that being said, even though these archaea are in such extreme conditions, they typically don't have a cell wall. They have a cell wall being something that really helps the bacteria to survive in a plethora of different conditions. It helps them regulate their osmotic pressures and helps them regulate what comes in and comes out. And the archaea don't have that, and they're in these really crazy extreme conditions. It's that monolayer plasma membrane that helps them to overcome that. So we'll get into more detail on the chemistry specifics of that in chapter 4. Another thing that all living things have that we can see the basics of with the high power microscopes are the ribosomes. And we've talked about these a little bit previously. The fact that the prokaryotes have a smaller ribosome, the 70S, while the eukaryotes have an 80S ribosome. But they function just the same, kind of like the plasma membranes. Even if I have slight chemical differences, the function is the same. And so the ribosomes are functionally identical. I can take genetic information from humans, like the genetics for creating the protein insulin. I can insert that into a prokaryotic E. coli cell, and the E. coli will make the protein insulin because the ribosome doesn't care. The ribosome doesn't differentiate where RNA is coming from. It just sees RNA and makes protein from it. So that is one of the similarities between the cells. And then all the metabolic networks. Remember at the end of chapter 1 we were talking about some of the new frontiers for microbiology, and we talked about the proteome and the metabolome. The reason that our new frontiers are especially fruitful for microbiologists is that it's the same basic story that we see in the higher order organisms. So it's a great place to start trying to figure out those intricacies and just build upon that framework for the higher order organisms. So all the metabolic networks are very similar. Glycolysis, it's the same between micelles and bacterial cell. The Krebs cycle, it's the same. The electron transfer chain, it's the same. So the metabolic networks are very similar between all living things. Now there are some striking differences, and those things are definitely points of interest. They make us different from the prokaryotes. So some things that you're probably very familiar with are, the fact that the eukaryotes have the DNA house. They have the nucleus for the DNA. But remember, be very clear here, and we talked about this with case study 1. You don't have to have the DNA enclosed in the nucleus to be alive. The prokaryotes don't have a nucleus. Who cares? Having a nucleus is not a requirement of life. Having DNA is a requirement of life. And you don't have to have a nucleus for it to work the way it needs to work. So your prokaryotes, they don't have a nucleus. Okay. no deal. They are still very much alive because they still have DNA. We also see that our eukaryotes tend to be larger and more complex because they have those membrane-bound organelles and the division of labor. Your prokaryotes are smaller. There's a reason for that. They don't have the division of labor. They're having to deal with more things in that space without, a specific organelle that's going to take care of that job. And that means they need to be smaller so the diffusion times work out, the metabolic need met, that enough nutrients can be brought in through the limited plasma membrane space surface area to feed the needs of this cell. Some surface area considerations are things we'll get into more detail in later chapters. And the organelles. Remember, the ribosomes are a real gray area. There isn't concrete agreement across the discipline on what they are, so you will sometimes see them referred to as organelles. For our purposes, we're going to say that they are not membrane-closed organelles. Remember, it's a big wad of protein and RNA, so it's not this nice little circular thing that you see in your textbook. That's artistic license. It's a big wad of protein and RNA together. It still does a specific function. It has a job, but it's not membrane closed. The eukaryotes, they have that laundry list of organelles we discussed previously. And we can see a lot of this with the higher power scopes. So this is just a figure, close to figures in your book. Basic cell structure is noted here. Everything's got the plasma membrane or the cytoplasmic membrane. Some things have cell walls, some things don't. It's not a requirement of life. See it in a lot of prokaryotes, but we don't see it in the archaea. We don't see it in the mollicutes. There are a specific group of prokaryotes, bacteria, that don't have cell walls. The eukaryotes, a lot of them actually have cell walls too. Plants, fungi, a lot of the protists. So, cell wall, it's a give or take thing. It's not a requirement of life. Please do not confuse it with the cytoplasmic membrane. It is not the same thing. We also see with our prokaryotes, we see the DNA. There's some other fun things going on there with information flow that we'll talk about in here in just one second. And then you have your viruses. So just kind of talking about basic cell structures, let's talk about some basic things that lie outside of cell structure, the viruses. For our understanding and our definition of life, these things are not living. That doesn't mean they're not important. They are parasitic, they are significant agents of disease, and they have a built-in system for invading and hijacking host cells. Now, a couple of things that's very interesting about that when you talk about microbiology, obviously we care about the disease pathology, but we also care about the fact that there are circumstances where we might want something to invade and hijack a cell. What if that host cell that we're talking about is a cancerous cell, and we want to put a horse into it, tell it to self-destruct? Viruses already know how to get in there. That's one thing that a lot of scientists are looking at from a cure standpoint. Also, people who have faulty genetics, maybe they don't make a protein like they should, and we would like to insert that gene back into their cells. What if somebody's pancreas has gone bad and not making insulin anymore? Well, what if we could go in there and tell them to start making insulin again? How could we get into that cell? Viruses already know how to do it, so that's a very fruitful area of research right now. The reason that these viruses have to invade and hijack host cells is that they lack their own protein synthesis machinery, things like ribosomes. They don't have it. They don't have it because they don't have the space to hold it. Some important things that we see with invading and hijacking cells is, does that necessarily have to lead to disease? And if you think about it, there's lots of viruses that get into us that don't ever cause us to be symptomatic. If you don't have symptoms, you don't have a disease. Not every bacteria you have in you causes disease. Every virus you have in you causes you a disease. About 4% of the human genome is viral DNA from viruses that has been incorporated into our genome and just has stuck around. It didn't hurt us. It just is there. And so don't confuse this invasion with always having to be disease related. Yes, the ones that we care about do cause diseases. That's why we care about them. The one that gets there and does nothing, we don't see it. We don't even know we should be concerned with it. But that doesn't change the fact that it's there. They're still there. There are different types of viruses. The viruses tend to be classified based on the extent of their protein casing. There are naked viruses, which are only some kind of nucleic acid with a protein case around the outside of it. Pretty stripped down model. There are other viruses that have the nucleic acid. They have a protein capsule around them, and then they usually steal some of the host membrane, and this is a cloaking device. This allows that virus to slide under the radar. And, when the host's immune system comes around, the virus says, hey, I'm you. You don't have to worry about me. You don't have to attack me. And it's a very clever mechanism for getting around attack. Now, there are some new things coming out that we're discovering that do some interesting things, like the viroids don't even have a protein coat. These are free-floating RNAs without protein coats. And remember, the way we figure things out in science is to try to break it and see if we can then, put it back together. And so when trying to figure out viruses, because most viruses are either naked or enveloped, the common way to break those, since they're enclosed in a protein case, is to treat them with something called a protease. A protease is something that destroys proteins. So this is the common mechanism for breaking things in the viral world. Well, there were some agriculturalists who were studying potato diseases and wine diseases, and so they suspected they were viruses. They did all those Koch's postulates things, and they didn't get a pure culture. They couldn't get these things to grow in culture, and so they went, hey, maybe this is a virus. So the common thing to do with the virus is, well, let's hit it with a protease and see if it loses its infectability. Well, proteases didn't touch these things. They were really confused. They just start throwing a kitchen sink at them to see, what can I hit these with that will kill them? And eventually they figured out if they just treated them with an RNase, which is a protein that cuts up RNA, that was enough to inactivate the virus. So that's viroids, which are free-floating RNAs. An interesting kind of step into the genetic information flow and phylogeny is we look at viruses and they are not on the phylogenetic tree. It's kind of a trick question. If I were to ask you, where do viruses fall into the phylogenetic tree? You would say, well, they don't fall into the phylogenetic tree. They're not living. The tree of life, part of the name is life. So if you have something not living, it can't go on the tree of life. That kind of goes back to our quizzes in class. Those 3 questions about with the scientific names of the organisms you can take to the bank. I'm never going to ask for a virus on that part of the quiz. They don't have scientific names because scientific names are reserved for things that are living and viruses are not living. So how do viruses tie in to our phylogeny? Well, the most common or the most current idea about viruses, especially the ones like viroids, while they are not on the tree of life, they